Hi, and welcome to 9.2 transport. If you want to stop it and have a read. Okay, so translocation is the movement of organic solutes around a plant. In the picture here, you can see the xylem. So the xylem moves water up the stem of the plant, and the phloem here is moving organic solutes, in this case sucrose, the red dots, down the phloem. Now, in the phloem, the solutes can move up or down. In this diagram, it just so happens to be down. The sucrose is being produced here in the leaf cell. Now, the sucrose has to be produced somewhere where there's lots of chloroplasts, which is generally in the leaf. Now, we call the areas that sucrose is produced the sources. The sucrose then moves into the phloem and moves towards something called the sink. Now, the sink is the area in the plant that's going to use the sucrose. So it's going to break the sucrose down into glucose, use that glucose for respiration in order to produce energy. So the source produces the sucrose, the sink is going to use it up. We've got all of that. So the phloem structure, the phloem is composed of sieve tubes separated by sieve plates. So these are the sieve plates here. You can see that there's some perforations or holes within these sieve plates. The reason for the name of sieve is because it's got holes like a sieve and only some things can get through. The sieve tube cells are also associated with something called companion cells. So this is a companion cell here, and this is a companion cell here. Now, these cells contain the majority of organelles that are needed for both the companion cell and the sieve tube cells. So the companion cells do all of the metabolic work for themselves and the sieve tube cells. So the sieve tube cells just need to concentrate on moving these organic solutes around the plant. This is a really nice picture actually of the phloem cell and a companion cell. So you can see all of the organelles within the companion cell, and the phloem doesn't have any of those organelles. Okay, so the sucrose has to be transported into the xylem, and we call this phloem loading, so the loading of the sucrose into the xylem. Now, what happens in this diagram here, you've got the apoplast, which is the cell wall, and then you've got your phloem here, is the hydrogen ions are actively transported out into the cell wall so that a high concentration builds up outside of the phloem. Then what happens is the hydrogen ions can move down their concentration gradient through this co-transporter, and the co-transporter co-transports the hydrogen ions with the sucrose. So the sucrose can't move through that co-transporter without the hydrogen ions. Therefore, this active transport must take place in order for both the hydrogen and the sucrose to be able to move into the phloem. In some species, there are kind of holes between the cells called plasmodesmata, and they allow the sucrose to move through into the phloem, but that's not in all species, just in some species. Okay, so within the phloem, there's quite high pressure, and the pressure then allows that, well, not high, but there's a high pressure at one end and there's a low pressure at another end. So that means that the solutes and the water is able to move from an area of high pressure to an area of low pressure. So one end of the phloem will always have a higher pressure than the other end. So if we have a look here, we've got this here, which is your water potential. Now, the water potential is the tendency of water to move from one area to another. So water will also move from an area of higher water potential to lower water potential. The solute potential changes depending on the amount of solute. So obviously, if you've got more solute, you're going to have a higher solute potential. If you've got less solute, you're going to have a lower solute potential. And the pressure potential is the pressure exerted by the rigid phloem walls. So phloem doesn't have xylem in its walls doesn't have lignin in its walls like xylem does. The walls of the phloem are already rigid, and that rigidity allows the pressure to build up because the walls don't move. Okay, aphid stylets. This has come up in a few exam questions. Now, an aphid is like a small green fly. This is an aphid here. And the stylet is like a sharp mouth part here. Now, what the aphid does is the aphid lands on the stem of a plant and it plunges that stylet into the plant stem. And this here would be the phloem. So what happens once that stylet moves into the phloem is then, as a result of the pressure, the phloem can then move into the stylet and the aphid can take in all of the sucrose that's within the phloem. So we refer to the water with the solutes in the phloem as cell sap. So that sap can then move into the aphid. Now that's got quite a high sugar content, so therefore it can be broken down and used to produce energy within the aphid. Now what scientists did was they allowed aphids to plunge their stylet into the phloem, and then they anaesthetized the aphids and separated them from the stylet so that all that was left was the stylet and the aphid is gone. What they then saw was that the sap continues to flow out of the stylet, which proves that there is pressure within the phloem. They also saw that at different areas of the stem, the phloem flowed at a different rate. So where there's areas of higher pressure within the phloem, the sap would flow out at a higher rate than where there is lower pressure within the phloem. Finally, we've got some diagrams that you need to know and you need to be able to identify. So we've got a cross section through a root. You need to know that this X within the middle is the xylem, and these bits around the outside of the X are the phloem. In a stem, you will have seen this in the 9.1 video, you've got the phloem here, and then you've got the xylem in the middle there, the red bits. This is the xylem and phloem in a leaf. You don't need to know this one. And then you've got xylem and phloem in a dicot stem and a monocot stem. So the dicot stem is this diagram here. And then the monocot stem, you can see that the vascular bundles tend to be dotted around the stem in a less orderly fashion than in the dicot stem. So you would know that this is a monocot stem because it's not in this kind of a ring pattern. They're all dotted around.